Okay, so we saw, for example, if we use beads that have fixed size holes in them that we can separate on the basis of size. What if we modify beads in other ways? Well, one way we could think about would be, what if we made a bead that instead of putting holes in it, we attached molecules that had charges on them? So for example, if I made a bead that had molecules with negative charges on them, when I pass through a mixture of proteins through that column, those proteins that are positively charged will be attracted to the negatively charged molecules on the beads, and they will stick. Whereas molecules that have no charge or that are negatively charged will not be retained, and they will pass through the column quickly. So what I've described there is something called ion exchange chromatography, in which I use ions as a way, the charge interactions as a way of separating molecules. If I am using cation exchange chromatography, which is what I've just described to you, the positively charged cations, the proteins that are positively charged, will stick to the negatively charged beads. If I use anion exchange chromatography, then I have the opposite design. I have beads that have a positive charge, and only the negatively charged proteins will be attracted to them, and all the other proteins will come out. Well, there's a third type of bead and a third type of chromatography that we use that's called affinity chromatography. And I'm going to show you a specific example of that in a second, but I'll describe one to you first. Imagine that I have beads that instead of putting holes in them, or instead of putting charges on them, that I put a specific molecule onto them. So let's imagine I took a bead and I attached to that bead a molecule of ATP. And for each bead, I put hundreds of those ATPs just sticking out everywhere from that bead. What would happen if I took a mixture of proteins from a cell and I added it to the top of the column and allowed it to go through the column? Well, what would actually happen is I would see that proteins that bound to ATP, maybe proteins that used ATP for their function, would grab a hold of that ATP on the bead and they would be stuck there. Whereas proteins that don't use ATP as part of their action would have nothing to bind to and they would pass through the column. Well, I would have just at that point separated proteins on the basis of their affinity for ATP, that is their tendency to bind to ATP. Well, that turns out to be a really useful function if I know something that a protein will bind to. And I can use it for a variety of things. I might want proteins that bind to GTP, or I might want proteins that bind to sodium, or you name it. Depending upon what I attach to that individual bead will determine what proteins are bound to it, and other proteins that don't bind to that particular atom or molecule will pass right on through. Well, a slight modification of that affinity chromatography is done in what's using histidine tagging. So I need to spend a little bit of time to explain that to you. Let's imagine, if you will, that I have a protein that I'm interested in making a lot of, okay? So this might be a version of human insulin, for example. And in this human insulin, I know I want to make a lot because I'm a drug company and I want to use a bacterium, for example, to make it cheaply, but then once I've made it, I've got to purify it so that I can give it to people to use for medicine. Well, how would I do that? Well, there's quite a wide variety of ways of getting a cell to make something. First of all, I'd have to know the amino acid sequence of the protein that I want. I'd have to have the gene, the DNA coding for that to be available. I'd have to put it into a cell. I'd have to have the cell make that protein by transcription and translation. And then I'd have to bust open the cell. So I'm at the point where now I've done all the various things that I need to. I would now want to purify this protein, okay? Well, there's a hard way to do that and there's an easy way to do that. And as I say, biochemists are lazy people, okay? I'm a biochemist, I can say that. I'm a lazy person. I want to purify this protein in the easiest possible way. Well, one would be to do traditional isolation, purification techniques that are very involved and are very expensive and so forth. And I could purify and I could get that protein that I was after that process. But there's also a means of modifying that protein so that I make it easy to purify, okay? And that's what I do in many cases. So instead of starting with the exact protein, I take a modified form of that protein, and I use that modified form in my purification system. And that's where this phenomenon called histidine tagging comes into place. So let's understand how that process occurs. Let's imagine that I've got a gene for a given protein. And there's my gene of interest, okay? And I take that gene and I modify it slightly. And the slight modification that I make is, here's my gene, but at the 5' end of where that gene coding would start, I put in coding for the amino acid histidine. And I put in coding for several of these histidines, typically 6 or 8 of these histidines that would be there. So that when this guy is made by the cell, it makes the gene, whereas if this guy is made by the cell, it makes the gene linked to histidine. There's a difference. This guy is just the gene, this is the gene plus this numerous histidines that are on the end. Well, why do I do this? Well, it turns out that these histidines, when they're linked like this, have a high affinity for an atom of nickel. And that's what I'm going to show you in the next slide. Okay, now this figure depicts what I've just described to you in words, and it assumes that we've already gone through that process. So we've taken, for example, and we've made our gene that contains this histidine tag that's on the end, and I now have a column that has those little beads, and instead of being attached to ATP, in this case they're attached to atoms of nickel. Well, what's going to happen? Any genes, any proteins that are made in this cell that have those histidine tags on them are going to be loaded onto the column and bind to nickel, whereas all those proteins that don't have histidines on them are going to go into the column and they're going to pass through it. So in essence, I will have separated the proteins that bind to nickel from those that don't. Well, it turns out that there are virtually no cells in the protein that start with a histidine chain like what I've described to you. So that almost all of the proteins of the cell are going to pass through the column, and the column is going to be bound by those proteins that have the histidine tag, specifically the one that I'm interested in. Now, there are a variety of ways of getting that tagged protein off of this column. One way would be to simply add nickel, in which case the protein would just grab the nickel and come off of the column, as it were. And the other would be to add a derivative of histidine, which does the binding to that column, called imidazole, and that would then bind to the nickel that's on here and displace the histidine tag protein, and our protein would be released. In either event, it doesn't really matter. What we end up with is our protein in solution over here, separate from all of the cellular proteins that are over here. So histidine tagging is a very powerful way for us to separate a known protein from other proteins.